Hello students, welcome back to our channel Food Tech Online. So today will be our fifth class on important questions of GATE Excel Food Technology subject. So all the aspirants who are sitting for the GATE Excel examination this year must watch this video till end. Foodborne bacteria Pseudomonas fragi. Options are pH 5, pH 6, pH 7 and pH 8. So what is the minimum pH value for the growth of this foodborne bacteria? It's actually option number A, that is pH 5. So this Pseudomonas fragi, it's a gram-negative saccharophilic bacteria, and even this bacteria is responsible mainly for the spoilage of dairy products. So here my correct option will be option number A, that is pH 5. Next, number 2. Choose the odd one out from the following. Option A, betalains, B, flavonoids, C, anthocyanins and D, chlorophylls. So the correct option is option number D, that is chlorophyll. So these three options, that is betalains, flavonoids and anthocyanins, these three are the water soluble, or you can say lipid insoluble pigments, whereas option number D, that is chlorophyll, it is lipid soluble or a water insoluble pigment. So the odd one out is option number D, that is chlorophyll. Number 3. Minimum pH value for the growth of foodborne bacteria Lactococcus lactis. Option A 5.3, option B 6.3, option C 6.5 and option D 4.3. So the minimum pH value for the growth of Lactococcus lactis, it is option number D, that is 4.3. So Lactococcus lactis, actually it is a gram-positive bacteria that is widely used in the production of fermented dairy products, such as you can say cheese, yogurts, in milks. So my correct option is option number 3, that D, that is 4.3. Number 4. Minimum pH value for the growth of foodborne bacteria Aeromonas hydrophila. Option A 5, B 6, C 7 and D 8. So the correct option is option number B, that is 6. So this Aeromonas hydrophila, it is a gram-negative rod-shaped bacteria that is mainly found in the areas of warm climate. Even this bacteria can survive both in aerobic as well as in anaerobic conditions. So the minimum pH for this Aeromonas hydrophila bacteria is around 6, that is pH 6. 5. The anti-nutritional factor present in faba beans. Option A gossypol, option B vicin, option C cyanogen and option D curcumin. So what exactly is this anti-nutritional factor? So anti-nutritional factors are those substances which interfere with the absorption and utilization utilization of nutrients in the body. So write down in the comment box what is the botanical name of this faba beans. So here the correct answer is option number B, that is vicin. So gossypol. So this gossypol, it is the anti-nutritional factor which is present in cotton seeds. Cyanogen, it is a anti-nutritional factor which is present in linseeds, and curcumin, it is a phenolic compound which is present in the roots of the turmerics. So here the correct answer is vicin. 6. The parameters of the plants and animal tissues that are an inherent part of the tissues are referred to as option A intrinsic parameter, option B extrinsic parameter, option C natural parameter and option D none of these. So the correct answer is option number A, that is intrinsic parameter. So what are these intrinsic parameters? Intrinsic parameters, these are the pH, moisture content, these oxidation reduction potentials, nutrient content, antimicrobial constituents, biological structures. Whereas these extrinsic parameters, they can include like the temperature, relative humidity, atmospheric gases, pH, pressure, others. So here the correct answer is intrinsic parameter, option number A. Question number 7. The transminase is an example of option A hydrolase enzyme, option B oxidoreductase enzyme, option C ligase enzyme and option D transferase enzyme. The correct answer is option number D, that is transferase enzyme. So the function of this transferase enzyme, actually it transfers the functional group from one molecule to the another molecule. Whereas hydrolase enzyme, this hydrolase enzyme, it is a type of enzyme that catalyzes the hydrolysis of a chemical bond. That is the hydrolysis. It is a chemical reaction in which water is needed to break down a compound into more further simpler substances. Option B, that is oxidoreductase enzyme. It is a type of enzyme that catalyzes the transfer of the electron from one molecule to the another. And this ligase enzyme is an enzyme that catalyzes the joining. Actually, it helps in joining of two molecules to form a single new chemical bond. So here the correct answer is option number D, that is transferase enzyme. Number 8. Fumarase is an example of option A ligase enzyme, option B isomerase enzyme, option C lyase enzyme, and option D hydrolase enzyme. So the correct option is option number C, that is lyase enzyme. So this lyase are the enzymes that catalyzes the cleavage of a chemical bond by the means other than hydrolysis or the oxidation. Whereas this isomerase enzyme it is a type of enzyme that catalyzes the conversion of a molecule from one isomer to another isomer. And this ligase and hydrolase enzyme just now we discussed. So here the correct option is option number C, that is lyase enzyme. Next. 9. Pyruvate carboxylase is an example of option A ligase enzyme, option B isomerase enzyme, option C lyase enzyme, and option D hydrolase enzyme. So the correct option is option number A, that is ligase enzyme. So just now we discussed that ligase enzyme, it is an enzyme that catalyzes the joining of two molecules by forming a new chemical bond. So here the option number A is correct. Number 10. Several yeasts can be used in the fermentation, but the most commonly known species of genus are option A Streptococcus, option B Lactobacillus, option C Leuconostoc, and option D Saccharomyces species. So there the correct option is option number D, that is Saccharomyces species. So we all know that Saccharomyces cerevisiae, it is also known as brewer's yeast, and it is most common yeast which is used in baking industries. Even other species of Saccharomyces like Saccharomyces bayanus and Saccharomyces pastorinus, these are also used. So the most common is the Saccharomyces species. Option number D is correct. 11. The deteriorative changes originating from within the food system is known as option A proteolysis, option B hydrolysis, option C putrefaction, and option D autolysis. So the correct option is option number D, that is autolysis. So actually autolysis, it is a process in which the cells or the tissues is broken down by its own enzymes, whereas the putrefaction it is a process of decomposing organic matter that is usually by the bacterial actions which results in the formation of other substances. And this hydrolysis, it is a chemical reactions in which a water is used to break down a compound into more smaller or simpler substances. And this proteolysis, it is the breakdown of the protein into small polypeptides of amino acids by the actions of enzymes, which is the protease enzyme. So with the help of protease enzyme, the protein is broken down into more simpler substances. So here the correct option is option number D, that is autolysis. Number 12. Generalization is more reliable than option A filtration, option B pasteurization, option C blanching, and option D none of these. So the correct answer is option number B, that is pasteurization. So generalization is more reliable than pasteurization. Actually generalization means sterilization. So in sterilization process what we have seen that it kills or eliminates all the forms of microbial life that includes the spores, whereas in pasteurization the spores are not eliminated. Whereas in blanching process we have seen that in this process it is immersed in the hot water or steam, and then it is followed by the cold water or a rapid cooling in the ice water. So it is actually done to eliminate to inactivate the enzymes. So here the correct option will be option number B, that is pasteurization. Next. Number 13. Which of the following is anti-ripening agent? Option A 2,4,5-trichlorophenoxyacetic acid, option B maleic hydrazide, option C sulfur dioxide, and option D sodium benzoate. So the correct option is option number C, that is sulfur dioxide. So what exactly is this anti-ripening agent? So anti-ripening agents are those substances which are used to delay, these are used to delay in the ripenings of the fruits or the vegetables. So among the following option number C, it is correct, that is sulfur dioxide. Even sulfur dioxide, it is a colorless gas that is used as a preservative in fruits and vegetables. Even in dried fruits, beers, wines, even it works by inhibiting the growth of the bacteria, fungi that can cause or delay the that can cause the spoilage, or it can degrade the fruits if it is not used. So, and next, this 2,4,5 trichlorophenoxyacetic acid, it is a synthetic auxin, and this chlorophenoxyacetic acid, it is a herbicide that was used to defoliate the broad-leaved plants. And malic hydrazide, it is a plant growth regulator which reduces the growth through preventing cell divisions but not the cell enlargement. And this sodium benzoate, it acts as a food preservative, which is mainly used in the acidic fruits like say pickles or in sauces, carbonate drinks, etc. So here the correct option is option number C, that is sulfur dioxide. Number 14. The breakdown of fatty material is referred to as option A autolysis, option B lipolysis, option C proteolysis, and option D none of the above. So the correct answer is option number B, that is lipolysis. So the breakdown of the fatty material is referred to as lipolysis, and this lipolysis, it is a process in which the triglycerides are hydrolyzed into glycerols and free fatty acids, and this process is catalyzed by an enzyme which is known as the lipase enzyme. So here the correct answer is option number B, that is lipolysis. Even autolysis and proteolysis we have just discussed what are these processes. So next, number 15. The breakdown of proteinaceous material is referred to as option A autolysis, option B lipolysis, option C proteolysis, and option D none of these. So the correct answer is option number C, that is proteolysis. So what exactly is this proteolysis? Proteolysis we have just discussed that it is a process by which proteins are broken down into smaller peptides or amino acids by the action of an enzyme. That enzyme, it is proteases enzyme. So here the correct answer is option number C, that is proteolysis. Number 16. Many reactions similar to oxidative rancidity during irradiations have been observed in option A water, B carbohydrate, C lipids, D none of the options. So many reactions which are similar to oxidative rancidity have been observed in lipids during irradiations. Actually, this irradiations, it can cause the formation of free radicals in lipids, and which can further lead to the oxidation of the lipids and form the off flavor or the off odors. Even this irradiations can also cause the changes in the fatty acid compositions, which can affect the nutritional value of the product. So the correct option is option number C, that is lipids. Number 17. The mechanism of food intake regulation is known as option A glucostatic regulation, option B lipostatic regulation, option C aminostatic regulation, and option D all of these. So the correct answer is option number D, that is all of the above. 18. The function of acid in food is to option A intensity and modify the test, B regulating acidity alkalinity, C act as microbial inhibitor, D all of the above. Correct option is option number D, that is all of the above. Few example of this food acids can include vinegar acids, tartaric acid, malic acids, or folic acid, fumaric acids, lactic acids, and others. So next. Number 19. The severe diarrhea and dehydration in human is due to the lack of option A renin, option B lysine, option C lactase, and option D all of the above. So the correct option is option number C, that is lactase. So however, the lack of lactase, there are enzyme that can break down the lactose. Lactose actually, it is a sugar found in milk or other dairy products, which can lead to the diarrhea or dehydration in some of the humans. So here the correct option is option number C, that is lactase. Number 20. Consistency and stability of dose is measured by A farinograph, B micrograph, C compress meter, and D all of the above. So the correct option is option number A, that is farinograph. Whereas this micrograph, it is a photograph or a digital image which is taken through a microscope or other similar devices that is used to show a magnified image of a small object. Whereas compressed meter, it is a device which is used to measure the flow rate of the compressed air. It is mainly used in the industrial areas mainly. So the correct option is option number A, that is pharenograph. 21. To prevent deterioration of food from microorganisms, which one of the following preservatives are used? Option A, NaCl, option B, sulfur dioxide, option C, nitrates, and option D, all of the above. The correct option is option number D, that is all of the above. So, to prevent the deterioration of food from the microorganisms, preservatives are added to the food products. So, these preservatives or these food additives actually, they play an important role in making the foods last longer, or it tastes better. So, especially these preservatives are used to control the microbial actions which has been taking in the food products. So, they provide protections from the spoilage from the microorganisms, so which includes like this sulfur dioxide. It is mainly used to preserve the dried fruits, wines, beers. And sodium chlorides, these are used as to preserve the meats, fish. Whereas this nitrates are used to preserve the meats. Even it contributes a pinkish color to the meat. So here the correct option is all of the above. 22. In preparation of fruit juices, pickles, and carbonated beverages, the preservative used is option A, propionic acid, option B, benzoic acid, option C, parabens, and option D, all of the above. So the correct answer is option number B, that is benzoic acids, and it's salts of benzoates, that is sodium benzoates. So these preservatives are effective against yeasts, bacteria, and are commonly used in acidic fruits, say like in tomatoes, sauces, pickles, carbonated beverages, sauerkrauts. So here the correct option will be option number B, that is benzoic acid. Number 23. Which fatty acid is present in rancid of butter? Capric acid, option B, salicylic acid, option C, butyric acid, and option D, palmitic acid. So when butter goes rancid, butyric acid is released from the glycerides of hydrolysis. So the correct option will be option number C, that is hydrolysis. Actually, this butyric acid, it is a short carbon chain acid that is responsible for the characteristic rancid or the rancid flavor in the butter. So the correct option will be option number C, that is butyric acid. Number 24. Osmotic pressure of the body fluid is maintained by option A, calcium, option B, sodium, option C, chlorine, and option D, magnesium. So the osmotic pressure of the body fluid is maintained by sodium. It is actually maintained by all like sodium, calcium, chlorine, magnesium, but the major function by the sodium here. So here the correct option will be option number B, that is sodium. Even chlorine is also important ion, but it does not directly play any important role in the maintenance of the osmotic pressure in the body. Number 25. Riboflavin exists in the form of which coenzymes? Option A, NAD, option B, NADP, option C, FAD, and option D, none of these. So the correct option will be option number C, that is FAD. So riboflavin actually it exists in the form of FMN, that is flavin mononucleotide and flavin adenine dinucleotide, FAD coenzymes. Whereas these coenzymes also they involved in a variety of flavoproteins enzyme reactions, which includes those that affects the energy nutrients in the citric acid cycle and in the electron transport system. Whereas this NAD and NADP, NAD it stands for nicotinamide adenine dinucleotide, and NADP it stands for nicotinamide adenine dinucleotide phosphates. These two are the coenzymes which are used in the cellular metabolisms. So here the correct option for this riboflavin coenzyme will be option number C, that is FAD. So with this we have completed our 25 question answers of GATE food technology subjects. So hope you liked this session. So do subscribe our YouTube channel and other social media links which are given in the description box. So till then, thank you.